Okay, hi everyone. I'm very happy to host this webinar today. My name is Olga Tarkhanova and I'm CEO of Chemspace. Before we start our event, I would like to remind you like a few rules we have for our webinars. So first of all attendees will be muted for the whole time of the webinar. The webinar will be recorded and you can kind of get access to the record on our YouTube channel, Chemspace YouTube channel. All attendees may have opportunity to submit questions in the Q&A session. And then once our presenters present their talks, we will kind of answer these questions. Okay, if you want about how we will structure our webinar, I will start showing you a few slides and talking about a little bit about Chemspace and what we are doing and how we actually connected to our presenters, to AlphaMa company. And then I will introduce the speakers and give the stage to the speakers. I hope it sounds good. And now I will share my screen and start with kind of a few facts, a few reminders about Chemspace. Okay. Okay. Yeah. Let's start with the first slide. Chemspace, a hopefully well-known platform, one-stop shop for the researchers where they can get any molecules they need, like small molecules, large molecules for their research projects. But apart from them, Chemspace also assist our customers with the first, actually, very important stage in any drug discovery campaign, hit identification. We have Inamine as our partner, and actually Inamine creates and market very well-known product, which is called Inamine Real. And Inamine Real, the size of Inamine Real space is now 2.7 trillions of molecules. So, and what is the goal, actually, our partnership is to develop a different approaches which we can use to explore this chemical space and accomplish this first stage of hit identification. If you watch about Inamine Real space, so Inamine Real space is a huge space, and the main idea here is to have a list of symptoms, kind of reagents, a list of well-validated chemical reactions. So combine these two components, we can get a really huge chemical space. And the compounds from this space can be delivered very fastly. The synthesis of these compounds can be done with very high success rate. And more than that, these compounds can be synthesized at very competitive pricing. So what we did, we developed like a few approaches how we can explore this space and how we can accomplish hit identification stage using Inamine Real. The first approach is called with synthesis. This approach was published in Nature a few years ago, and the main idea here to perform gigascale virtual docking of such a huge chemical space. The next approach is called crystal structure first approach. And here we're dealing actually with fragment-based drug discovery. When we start with fragment screening, identify hit fragments, and then we use Inamine Real to grow the fragment like very quickly and very efficiently. Once again, using Inamine Real reactions. And the third one approach is called the AlphaMa approach. And the idea here to screen a DNA-encoded library and use this information to build machine learning model and apply this model to, once again, Inamine Real space. So here, the workflow in details. So the classical workflow for DNA-encoded libraries, we start with the screening, then we do an analysis of the data we got from screening, we perform amplification and sequencing, and ended up with some number of off-DNA hits. And this approach works great, and we have like a lot of kind of confirmations, published work, and we also have a few drug candidates in clinical studies right now. What we suggested to do, we suggested to have like additional workflow here when we can use the data out of DEL for machine learning. And since the DEL library is very huge, I would say, it's a perfect alternative to high throughput screening, because a DNA-encoded library can contain like from millions to billions number of compounds just in one tube, and generate a really big set of data. And this is a perfect fit for machine learning, actually. And when we build the machine learning, we can very easily use this machine learning model to predict the potential hits out of chemically accessible spaces like enamine real. And of course, today we're actually here to discuss the DELT technology in detail, to explore, to find out more information about DEL libraries, how they created, which libraries are accessible off the shelf, which libraries can be, let's say, synthesized per request. It could be custom DEL libraries. And I'm very proud to introduce two our speakers today. Both of them are present company which is called AlphaMa, and we have been working with AlphaMa for many years already. And I should say that it's a great partner. We successfully accomplished like a lot of commercial projects together. They provide us with their DEL libraries, and then actually these libraries are screened, and our role of Chemspace actually to use this information to build this machine learning model. Okay, the topic of today's webinar is intelligent approaches, applications of DNA-encoded chemical libraries. And the first speaker I'm honored to introduce you is Mei Chu. She's the head of DEL platform at AlphaMa. She got her PhD in Dresden, in Technical University Dresden, Germany. In 2022, she joined AlphaMa as the lead of DEL screening team. And since 2023, she is the head of DEL platform at AlphaMa. And the second speaker I'm honored to introduce you is Shun Wan. She is the director of discovery chemistry at AlphaMa. She got her PhD in organic chemistry in Shanghai Institute of Organic Chemistry. She worked for 2 years as associate scientist at Amgen, and then she joined Shanghai Institute of Materia Medica as associate investigator. And quite recently, already in 2024, she joined AlphaMa and became a director of discovery chemistry at AlphaMa. So now I pass the word to Mei, and she will present the first part of the talk.
All right. So so do you see the screen now? Nice. Okay, so thank you, Olga. Thank you so much. We are very happy for this opportunity to share what we are doing with DNA-encoded chemical libraries at AlphaMa. So we are not going through all the histories of this DNA-encoded chemical library technology, or DEL, because in my humble opinion, so DEL is no longer something new to whoever interested in small molecule drug discovery. So I will just spend one slide to briefly introduce the concept of DEL and how a DEL is generally synthesized. And to align with that, we'll also show our approaches to provide on-demand DEL based on individual customer needs, because we believe that an excellent technology should be readily accessible to whoever interested and to support or to strengthen their platform. Then we'll also talk about how we apply DEL more than just a hit finding. So we will be covering how we use the focused DELs to optimize small molecular hits, and how we identify covalent hits from covalent DELs, and also how we explore druggable chemical space beyond the rule of 5 using macrocyclic structures. And at the end of the talk, we'll also introduce what we offer at IDEL platform at AlphaMa. So, as the name indicates, DNA-encoded chemical library is a collection of chemical compounds. So here I'm trying to use different shapes to show different building blocks, because those compounds consist of different building blocks. And I'm trying to use different color to show different structures, so to show the diversity. So in DEL, what's special is that the compounds are linked to a DNA sequence. So each compound is linked to a unique DNA sequence, which can then be unambiguously identified by high-throughput sequencing. So another nature is that when the compounds are linked to DNA, they are soluble, water soluble, and millions of those compounds can be dissolved in water, and then they can be stored in a simple epitope, as Olga just mentioned. So being one of the best examples of combinatorial chemistry, the DEL, we also synthesize DEL by using the split and pool synthesis. So to start with, we have a batch of building block 1, and each different building block 1 is conjugated to a unique DNA sequence by DNA-compatible chemical reactions. What we get is the DNA compound conjugate. So the individual conjugates then are mixed together in one solution. Then this by pooling, so then this mixed solution is split into new reaction vessels, and the number of the new reaction vessels is determined by the number of second building blocks. Now the cycle 2 synthesis starts. To this pooled conjugate, we undergo the second reaction with the second building block. And after individual chemical reaction in each independent vessel, then the encoding for the second building block will follow by DNA ligation. So pooling, splitting, reaction, encoding can be iterated to generate a library of a tremendously huge size in a time-efficient way. So at AlphaMa, we are also dedicated to providing DNA-encoded libraries based on their individual needs. And what determines a DNA-encoded library is basically two things. One is the building blocks, and the second is the chemical reactions to join the building blocks together. So nowadays, abundant reagents are specially curated for DELs. So there are readily available building blocks that are provided by global vendors such as Inamin. And in practice, we like to categorize them based on their functional groups, like monofunctional building block, bifunctional, and trifunctional, depending on the number of available reactive handles. So for monofunctional reagents, they can be used as the last cycle of a library, while bifunctional reagents or building blocks, they allow orthogonal reactions with two different reactive handles, and they can be incorporated in any cycle of the library synthesis. And trifunctional reagents usually accommodate 3 different reactions and therefore they often serve as a scaffold to build a branched construct. And what's more intriguing is that we have the chemical toolbox to convert one functional group to another. So for example, this amino group, the building blocks within amino group can undergo amine acylation, reductive amination, and so on. But they can also undergo cyclization after we convert it to outside. So this dramatically increases the utility of the building blocks. The same building blocks can be used for different reactions and therefore for different design of libraries. So building blocks are the fundamental and most important ingredients for a library. And the recipes to cook those ingredients are the chemical reactions. So now there are many or numerous pre-established DNA-compatible chemical reactions that allows the CC bond formation, CN bond, and also other bonds, as well as cyclization, which is very important for drug-like properties. So the building blocks and the reactions together can form such an interactive dynamic reaction network, and we can choose any of the paths to synthesize library of your design. And apart from those pre-validated or well-established reactions, we are keeping exploring new DNA-compatible reactions to expand the druggable chemical space covered by DELT. And meanwhile, we put a special focus on heterocyclic structures because they are ubiquitous in approved small molecule drugs or biologically functional chemical entities. So if you are interested in building a DELT incorporating those important scaffold, we are able to design, optimize, and establish the synthetic routes. So again, we are committed to providing DNA-encoded libraries to strengthen your platform, and we can assist you on 3 aspects. First of all, if you have your interest of idea of reactions, scaffolds, or fragments, then we are able to provide a library design about how the compounds will look like and how the compounds will be made through a series of chemical reactions. Then this will automatically determine what kind of building blocks we should use. I mean, more specifically, what type of functional groups. So again, we have access to many different or vast collection of building blocks. And more importantly, before we initiate a library synthesis, we would validate each building block to make sure that we are using qualified building blocks with predictable product and also promising yield. And this validation usually goes hand in hand with reaction optimization in terms of reaction conditions like equivalence or temperature, catalyst, and so on. And during the library synthesis, we also apply strict QC strategy to monitor the progress of the synthesis. So this was all about building a DELT or customized DELT from scratch, from single building block, from cycle 1, and from individual reaction step. And there is an alternative to make a faster and cost-effective library is to use our in-house intermediate libraries. So our diverse intermediate libraries with reactive handles can be used to attach your fragments of interest and then can rapidly generate focused libraries. In this way, your fragments can be developed into a new level of affinity and also diversity. So in case your fragments of interest do not have a DNA-compatible reactive handle, our team of experts can help designing based on our knowledge in MedChem and also its binding mode. So speaking of focused libraries, next I would like to show 2 examples that how we utilized the focused libraries to get more SAR and more potent hits. And before that, I think I should go through briefly how a DELT screening works. So again, And the compounds are in a simple epitope, and our target is usually on a solid support via an affinity tag like his tag or biotin or flag tag of your choice. So this complex, then we throw this complex into the epitope, and after incubation, washing away those weak binders and bound molecules, we denature the protein to release the binders. And this released binders can be resubjected to the screenings that run multiple rounds to increase the signal-to-noise ratio. Finally, the elution are sequenced by high-throughput sequencing, and then the sequence information are translated into the identity of individual building block. The copy means how frequently the compound was binding. A higher copy obviously indicates higher potency in general. But of course, we need to more deeper analysis to exclude false positives or background noise. So those digits or the TXT files can be then easily visualized in a 3D plot so that we can identify SAR quickly. So by taking this cubic plot, for example, so this is a data from a three cycle libraries because we have building block 1, 2, and 3 on each axis. So when we analyze the data, we pay much attention to those lines because line means those compounds sitting on this line actually share the same building block 1 and building block 2 combination, while building block 3 can differ. And this is a strong indication about structural activity relationship. So we try to find compounds from those lines and resynthesize them off DNA and to validate them by biophysical or biochemical assays. So to start with our first case, our target was this tryptophan aspartate repeat domain containing the protein 5WDR5. This is an essential scaffold protein, and by binding to different partner proteins by this Win and Meek pocket, it can regulate the transcriptional level. So in this case, we were interested in identifying a weak pocket binder, which has been shown to be an effective strategy to treat acute leukemia. So with this target in hand, we started with a general DELT screening, and from a 3-cycle linear version library, we found these lines or SAR. So these two lines, meaning that they are the combinations of building block 2 and 3, and we found these two lines actually also share high structural similarity because the building block 3 are basically the same, and the two also have very high structural similarity. And this SAR was also nicely reproduced when we changed the tag from his tag to biotin tag, and also when we blocked the Meek pocket by a Meek peptide. And it's indicating that the compounds on these lines are not Meek binders. So starting from this SAR, we did some resynthesis work and validated them by homogeneous time-resolved fluorescence using the Win pocket binder as a probe. So if we see a dose response inhibition, then we get an idea that the compounds are actually binding to Win pocket. And as a result, we found this compound 5 was showing double-digit nanomolar IC50. And further molecular docking was suggesting that this 5-methyl-imidazole is binding in the center of the Win pocket, while the building block 2 in blue is involving in hydrogen bonding with 2 cysteine residues. And what's actually motivated us to build a focus library to further expand this compound is that actually there were no moiety of the compounds actually binding in the S4 site. So this motivated us to try to expand this compound somehow so that we can also conquer the S4 site. So since this SAR is telling us that this building block 2 and 3 combination is important, we kept this core structure unchanged. Then we build a focused DEL by verifying the first cycle using 124 amino acids as cycle 1. And we also incorporated a cycle 2 building blocks here because previously it was bromide, so which can be easily replaced by boric agents using Suzuki reaction. And this library has a size of 44,000, which is indeed a focus library, much smaller than previous general library. And this library is a two-cycle library, which we can visualize by 2D plots. And interestingly, we actually included this initial building block 1 in this 124 collections, and we found this one was the still most abundant reagent building block. Therefore, during the hit validation, we kept this building block 1 unchanged, and based on the enrichment signal, we altered the R1 part with different groups, and we obtained a single nanomolar binder. So this compound, actually so identified from the focus library, had actually some moderate anti-proliferative activity on cellular assays. So further medicinal chemical activities have removed this unnecessary chiral center. We also chopped off this DNA linking part, which is a formamide group, and also we reduced the number of hydrogen bonding acceptors and acceptors and donors. As a result, we ended up with a compound with single micromolar anti-proliferative activity, but single nanomolar IC50 at molecular level. Now the second case is also a collaboration project. We had a target, and we also had a known orthosteric inhibitor, which had poor pharmacokinetic properties. So with an aim to identify a new series of hit, we ran a general DELT screening. So in two conditions, one is with a target alone, and the second is target with excess orthosteric inhibitor to block the pocket. Then DELT screening outcome was showing that this building block 1 and 3 combination was not present in the second condition. And heat resynthesis and AS mass measurement has shown that this heat compounds has around 25% of potency compared to this initial inhibitor. Then based on this SAR, the structure, and also our knowledge about this pocket, we then redesigned 3 different focus libraries. Since this is a combination of BB1 and 3, we tried to diversify the linking part, so the BB2 part, with incorporating different scaffolds. Again, we screened these 3 focus libraries and in 3 different conditions with target first, and also target plus this orthosteric inhibitor. And then also we included another condition, which is target with the initial hit. And the enriched feature was showing that the compounds, so we found features that are present in target alone or target with initial hit, but not in the target plus orthosteric inhibitor. That indicates that the compounds can bind to the same pocket, but it has lower affinity compared to the original one, but stronger affinity compared to the initial the hit. And indeed, the off-DNA resynthesis and AS mass measurement has shown the improved potency, but still weaker than the initial inhibitor. So now currently we are in the progress of medicinal optimization to improve the property and also the potency. So to summarize, DELT is not just for hit finding. So if we have any knowledge about our fragment, and which can be maybe come from DELT or HTS or other technology, then we can further elaborate this fragment into a new DELT library. And this allows us to explore the vicinity of the binding pocket, and also the DELT data can tell us some positive or negative SAR. Okay, so I would like to hand over to my colleague, so she will continue sharing our applications in covalent DELTs and also macrocyclic DELTs. So now I would like to stop share, and Jen, you can take over.
Hi, can you hear me and see the slides? Okay.
Yes.
Yeah, okay. Hi everyone, I'm Xuan, and it's my honor of sharing other two products at AlphaMa in the second half of this report. The first one is a covalent DNA-encoded libraries. The covalent drugs have been used therapeutically for over a century, especially the EGFR inhibitor and the BTK inhibitor iluptinib. Both are the multi-billion dollar drugs. The covalent molecules can offer key advantages over the reversible ligand in their sustained target occupancy and the enhanced target potency and selectivity. Early examples of the covalent drugs generally originated serendipitously from the natural products or from the more rational CDD approach by incorporating the electrophilic warhead into a known reversible ligand. While the DNA-encoded library platform could provide an electrophile-centric screening platform, enable the direct identification of the covalent ligand from the outset because all of the library members had equipped with an electrophilic warhead. AlphaMa also provides diversified covalent library featuring the irreversible covalent and reversible covalent library according to the different electrophilic warhead. And we also have a mature and efficient selection workflow for the irreversible covalent libraries. Compared to the reversible selection protocol mentioned above, the major difference in the irreversible selection protocol mainly focus on the round of the bonding wash elution cycle. Reversible selection enables multiple rounds of selection to sufficiently enrich the binders over the background, while the irreversible selection can perform only one cycle due to the formed permanent bond between the binders and the target. Uh, This slide shows some covalent libraries in AlphaMa that have ready for selection with targets of interest. Our team have developed and validated various on-DNA chemistries and synthetic routes to prepare diversified covalent libraries, including both the linear and privileged scaffold. Furthermore, we have enriched experience combined with abundant available building blocks and other robust chemistry. We could design and prepare more valuable structures. This fragment could enable to offer key binding interactions with the target. We also validated diversified electrophilic warheads targeting different nucleophilic residues of the protein target, such as cysteine, lysine, and tyrosine. Here are some representative covalent compounds from our libraries. This is a published case in our team, presenting a commonly applied strategy at AlphaMa. We could initiate a project from a ready-made intermediate library. In this case, it is a trizine-based two-cycle intermediate library bearing a free imine group, which could connect with the electrophilic warhead to prepare a covalent library containing about 0.7 million library members. This library then selected with three targets, and the enriched feature was shown in the three-dimensional plots. The off-DNA synthesis and in vitro assay were performed to confirm these compounds. The truncated compound without the electrophilic warhead were also synthesized as comparison. The activities of these reference compound decreased significantly, indicating the importance of the electrophilic warhead. This slide is another published case. DELT could provide an efficient high-throughput screening platform, but if we want to improve the success rate of the selection, we would like to select specific target instead of randomly select protein targets. So we combine the activity-based proteomic profiling technique with our covalent DNA-encoded libraries. This chemical proteomics technique could help us to find the proteins sensitive to some specific residues. In this case, we use the sulfonyl fluoride warhead to targeting the tyrosine residue in the protein and select three targets for the further selection. And we design and produced the covalent library bearing three sulfonyl fluoride warhead. The DELT was then selected with these three compounds and the in vitro activity assay and the crystal structure validated hit compounds with the protein targets. In addition, other teams from both the academic institutes and the industry also applied the DELT platform to find covalent binders against a variety of target proteins, verifying the feasibility of these techniques in covalent drug discoveries. Moreover, the scientists from the Baylor and the GenTech use the DELT platform to find the reversible covalent ligand. The aldehyde in the building blocks could form the temporary bond with the cysteine residue in all the proteins. We can build such libraries with abundant aldehyde containing building blocks. This slide is a brief overview of the on-demand covalent DELT service at AlphaMa. Except for the covalent warhead, the fragment bound to the protein target also play an important role because it could help the warhead access to the right nucleophilic residue. AlphaMa has prepared a diversified intermediate library. If you have a specific ligand derived from other selection method or unknown ligand, we could link it to our ready-made intermediate library as well as the electrophilic warhead to prepare the final covalent libraries. We also could de novo design and prepare libraries according to your requirements of the characteristics of the ligand. We are flexible to the needs of the project. Okay, the next product at AlphaMa is our macrocyclic DNA-encoded libraries. The peptides has extensive application areas, including as therapeutic drugs, especially the orally bioavailable drugs such as MK0616. Or it could be used as a homing peptide in the peptide drug conjugate. The payload in the PDC could be the cytotoxic agent or the radionuclide. It also could be used in the diagnosis and imaging. Most of the active peptides originally from the naturally occurring products or their derivatives. But with the continuous advancements in the technologies, random design rational design uses the CDD and AIDD, or the serendipitous screening techniques, have already played a crucial role in current drug discovery. The powerful chemical tool in the DNA-encoded library enabled us to design and produce the macrocyclic libraries, and it has several advantages. First, it incorporated extensive variation in side chain using abundant non-canonical amino acid units. Second, it could produce diversified scaffold geometry while various cyclization method. Thirdly, rational library design allows us to offer both the small ring scaffold and the large ring scaffold, which could be used for different application scenarios. Both our group and the other research team have done extensive investigation in the field of macrocyclic libraries. This slide is a brief overview of our ready-made macrocyclic libraries at AlphaMa. AlphaMa cooperated with many vendor partners who could provide us with numerous amino acid regions, helping us to extend our the residue diversity of our libraries. Our team also dedicated to develop and validate efficient cyclization method, which could be used for the library construction. Moreover, we have designed and prepared two types of the macrocyclic libraries. The first series has a small ring scaffold, which is suitable for identify hate compounds with oral bioavailability potential. Another library series has a larger ring size and more variable binding sites. It is suitable to find the hate compounds that feature high binding affinities and could be used as a homing peptide in the PDC. Another important point is that the macrocyclic library could provide valuable and enrich structural binding relationships for us to perform the structural optimization. The histogram in this slide are the physical-chemical parameters of the small cyclic ring libraries. Most of the oral peptides have a molecular weight below 700, a polar surface smaller than 200, and 5 or fewer hydrogen bond donors. The parameters of the small ring scaffold library are close to this standard, and the polar NH bonds could be replaced by the N-methylated amino acid to decrease the number of hydrogen bond donors, as well as improve the overall lipophilicity, which could enhance their cell penetration. This is one of our published case, which we built a tetrameric cyclic library using a cell aeration cross-copying reaction. The formed cell area bond mimics the naturally occurring ether-bridged peptide scaffold. The formed scaffold is a small cycle ring containing two endocyclic amino acid variable unit and one exocyclic acyl capping site. The library was then screened against the protein P300, and two hit compounds was of DNA synthesis and in vitro and validated by the in vitro activity assays, giving single digital micromolar inhibitory activities. Moreover, we calculated their parameters by the professional software. Both of these compounds give a good overall bioavailable parameters. Our macrocyclic libraries also selected with a challenging PPI target and delivered many enriched features. Several enriched features were on DNA synthesis and validated in vitro assay, providing inhibitory activities in a range of nanomolar and micromolars. More protein targets, including the PPI target, tumor-related targets, and the virus target, were also screened with our macrocyclic libraries. Almost all the selection could provide authentic outcomes. The on-DNA of DNA synthesis and the in vitro activity validation is ongoing in our team. We also successfully incorporated the electrophilic warhead into the macrocyclic to prepare the covalent macrocyclic libraries. It also could provide an efficient method for screening and identifying covalent peptide library ligands. AlphaMa also provide on-demand microcyclic DELT service according to the requirement of the customers. If you have a specific ligand, it could derive from the known literature of other screening method. AlphaMa has experience to design and prepare the cyclic the linear peptide libraries based on this ligand, and the prepared focus library could then selected with the target to find hit compounds with improved potency. Next, the last is the published case. The scientists in the phylochem prepared two linear libraries based on a known FAP ligand. After the screening, validation, and structure optimization, the peptide coupled with radionucleotide lutetium to give a product with high potential inhibitory activities. Its analog OncoFAP23 has now is in the preclinical phase to treating the malignant tumors. The cyclic peptide also could conjugate with radionucleotides to use as radiopharmaceuticals to treating the tumors. And finally, I want to introduce the AlphaMa. AlphaMa is a new drug R&D company based on the DELT and AI technologies. The ideal platform of AlphaMa has a first-class DNA-encoded library technology and specializing in small molecular drug discovery, with its mutual screening and high success rates, providing global partners with cost-effective, efficient, and comprehensive one-stop DELT screening service. The AlphaMa team also have extensive experience with the covalent DELT and the macrocyclic DELTs. So if you have interest, kindly to contact us. This is the end of my report. Thanks for your attending.
Thank you very much. Thank you very much for both speakers. It's very interested talk, and actually I'm a big fan, and I really like the DELT technology. And actually, I saw the results from the projects we run together that the technology allow us to actually to find the pretty good hits after the DELT screen, and actually it's good to know and good to understand how we can use DELT technology also for hit expansion as well. I encourage everyone, if you have any questions to our speakers, please type them in Q&A section in Zoom. And meanwhile, I will probably start with questions I have. The first question actually about the readily available library, DELT library you have. And I know that you have like classical DELT library available for the purchase and also Covalent libraries. Yeah, could you please comment like on the size of these readily available libraries? And the next question is, you mentioned that you have so-called intermediate library, which can be, let's say, and the customer can actually provide some fragment or fragments he would like to add to this library. And how long it's going to take to, let's say, to transform this intermediate DELT to the final kind of customized DELT?
Thanks, Olga. So maybe I can answer the question. So yeah, about your first question, yeah, of course, we have general reversible DELT libraries, and currently, so we keep expanding our library set. So what I can tell you for sure is that we have over 100 libraries of different size, different chemistry, and different diversity. And so, but although I did not mention it in my slides, we actually only limit our cycles, split and pool cycles, up to 3. So we have 2-cycle and 3-cycle libraries. So now the diversity is over, of course, over 1 billion. I don't remember exact number, but yeah, it's over 1 billion library. And when we offer their screening service, so maybe you don't want to try all 1 billion compounds or 100 libraries. As if you want to understand your target, if you want to know whether my target is druggable, you can start with something, a very small kit, like 20 libraries, 30 libraries, because we are flexible about that. So we have flexible offers of different library size and also, again, and then different pricing for that. So if you're interested, you can also send us an email. And about your second question, so about the converting a fragment into libraries, this is very fast. So we already have pre-made intermediate libraries. And if your fragment already has a functional group, is a compatible functional group, then it only takes us 2 to 3 weeks to convert it to the new libraries.
Yeah, thank you very much. It's very helpful. And one more question for me. From me, it's actually about, you mentioned that you kind of the variety of libraries you have. It could be like 2-cycle and 3-cycle. Like, could you please kind of yeah, I understand the difference. Actually, that these 2-cycles libraries consist of 2 building blocks, and 3 cycles consist of 3 building blocks. Would you recommend to use like 2-cycles, 3 cycles, maybe combination? Like what is the best way how to combine this initial library for the project?
So based on my experience, after a lot of data analysis, after we screen 2-cycle and 3-cycle libraries, I cannot really say, okay, this target is more suitable for 3-cycle or the other way around, because although 2-cycle uses 2 rounds of iteration, uses 2 different building blocks, sometimes it can be bigger than a 3-cycle libraries when we include a constant scaffolding in the middle. So as I mentioned, like if we offer a 20 library kit or 30 library kit, we would try to include both 2 and 3-cycle libraries to see, or maybe your target has a preference. So we wouldn't say, okay, your target's protein, your pocket is very small, maybe you should try to go for 2-cycle only. No, we wouldn't say that. Yeah.
Okay, thank you very much for this answer. So we have an answer from the audience. Is it possible to create a custom DELT library from customers' in-house building block collection?
Definitely. So, but one condition is, again, so those building blocks should have a suitable functional group. That's the only precondition. Then if you ship us your in-house building blocks, we can, of course, definitely make a library for your purpose, for sure. Yeah.
Yeah, I see. Okay, and another question is about physiochemical parameters actually for your library, for your intermediate library, for your kind of readily available library. Like, do you calculate some physiochemical parameters for the compounds for these libraries? Like what range of like molecular weight, log P, you're kind of targeting?
Yes, we do. So when we calculate the parameters, for example, I think Xuan just showed a slide about that part. So for the library, like for one of the library, we randomly pick some thousands of different compounds, and then we calculated it by software to see the distribution. Yes, we do that. Mm-hmm.
This is actually for like a distribution for microcycle libraries. Yes, is it correct?
Mm-hmm. We do have actually a slide. We didn't put it here today, but we do have calculated for general libraries, for small molecule libraries, that it proves that they are in the scope of drug likeness in our based on our understanding about the drug. Yeah, they are in that scope.
Yeah.
And also we have yeah.
Yes, sorry for interrupting. Yeah, the next question is, how long does it take from selecting the library to having validated hit?
So usually screening, if we get a target from our customer, the screening will usually the whole screening will take around 5 to 7 weeks. But if you mean validated hits, it's a bit confused. So we can deliver a DELT screening report in 5 to 7 weeks. Then we need to synthesize the off-DNA compounds. This actually takes another like 3 to 4 weeks in general. Then afterwards, it depends on the assay, the available assays to validate it.
Yeah.
And
this is like a multi-step process actually to like between DELT and hit, validated hit, but yeah. Okay, the next question, can DELT be used for targeting protein-protein interactions and kind of complicated systems?
Yes.
yeah, definitely. We have done actually quite many such, yeah.
Sean,
Simply
go ahead.
Yes, our the macrocyclic library has screened many challenging PPI targets, and almost all the selection outcome could deliver the authentic enriched features. Yes, our the macrocyclic library could select with these targets.
What about small molecule libraries? Is it worth trying to use small molecule kind of DELT libraries to address these protein-protein interactions?
Also, I can add that to the question. So, yeah, we have experience with dealing with like a target protein complex during the screening. Like you have two different targets, so two targets binding together, and we screen it against the complex. We have experience with that, and also, as you asked, small molecule DELTs can also be used for screening. Yeah, but also what Xuan said is also correct.
Yeah, thank you. Okay, the next question is, could you suggest select DELT library based on pharmacophore model?
I'm not sure how should I understand the question.
Yeah, to be honest, yeah, I think the question probably is that if we have some kind of some kind of information about the ligand which could bind the target. Could we use this information to basically design or kind of pre-select some DELTs you have available?
Oh, thank you so much, Olga. Now I get the question. So similarly, we also had such a case, like we know there is some pharmacophore binding to this certain target. Then what we did is we have our DELT library, and also we have our enumerated the library corresponding to that DELT library. And then we also ran some docking, high-throughput docking, to see what types of compounds or what type of library would be suitable for this target. And based on this pre-information, then we went for the screening with certain library of our choice. And we found it delivering quite some hits. I think some information with the pharmacophore model would definitely help to determining our directions.
Yeah. Thank you. And another question. What are your go-to methods to validate compounds from a covalent DELT hit set? I mean, once we have like
DELT hit, we can run some covalent hit. We can go to methods to validate compounds. When we have some covalent hits from DELT screening, what we do usually is we resynthesize those hits on DNA, and then with those conjugate, we incubate it with the target, and then when we can run this drive shift assays, then you can see it when the protein is modified by the DNA, you see the different height. And we also have implemented this approach to prove our hits from covalent screening.
Would you like to add something, Sean? Okay. Okay, and a few other questions. How many hits do you typically get from the DELT screen? Does it depend on the size of the library?
To be honest, I think it depends more on the target. The SAR or the feature usually depends a lot on the druggability of the target. Let's say a target is druggable. The size matters. The size of the library. I would say not really. Based on my experience, not really. So I said you asked how many hits. So when we find hits, we carry these SARs like the lines we showed on the presentation, and yeah, we consider them as hits. But it's really different, and for every different target, we get very different results. So it's hard to say. But if we find some clear SARs, I would say this is a successful screening. Yeah.
Thank you. And the last question.
About the size of the library. Yeah. Please go ahead, Olga.
Yeah. One more question. Do you need to change the incubation time for the Covalent library? I mean, to optimize probably somehow time for incubation in case of covalent DELT.
Yeah, I can comment on that. Also, we typically run covalent screening with 1 hour. That's the same as a reversible screening. But if you have a known tool compound that you can conjugate it on DNA, then we can titrate the incubation time. And we do have this experience and test it. Sometimes then we prolong the incubation time to 4 hours. Yeah. So based on the tool compound, we can determine an incubation time then for real screening. Yes, we can run some pre-selection tests. Yeah.
Okay, and the last question, can you comment on a diversity of your libraries? For example, a 2D versus 3D shape.
3D shape. Chemistry.
Like how you define diversity for the libraries, what kind of methods you usually use, and whether you analyze this kind of 2D, 3D shape of the molecules.
We have been trying to analyze the diversity based on 2D and 3D shape. Like, if I understand correctly, 2D means we may not consider the chirality. Probably I'm wrong. And so, yeah, when we analyze when we try to draw a map about this chemical space, we have been trying to describe it with different descriptors, and both 2D and 3D. And we have tried to overlay the chemical space of our library and also the clinical drug candidates and also marketed drugs. And our library, they have some overlapping part, but our library is much broader compared to that. Yeah, approved drugs and clinical candidates.
Yeah, thank you. Like the question
and again,
sorry.
Oh no, you can go ahead.
Yeah, I'm saying that the questions are still coming, and the next one: how do you wash the Covalent library during selection?
We wash as we do for the reversible screening first, but then we also denature the protein to release those reversibly bind part. Then we keep the so the covalent hits should be conjugated covalently to the protein, and the protein is remaining on the bead, magnetic bead. So we took the whole beads as our template for the sample preparation.
Yeah. Okay.
So we include a heating step.
Okay, it's clear. Okay, I think we are done with the questions, and what I should do next, actually, to thank you a lot for being here today, for delivering like wonderful talks, to providing information about your company, your technology, like the services you can provide, how you can build your libraries, like variety of libraries, how you can be very flexible. And as I already mentioned, I really enjoy working with you guys. Thank you very much. Thank you very much for your talks. And everyone, thank you very much for your attention.
Thank you very much to you too.
Thank you.
Thank you. Thank you very much.